Hello friends, welcome back to Varsha's videos. In this video I'll talk about golden rice. Introducing rice. Golden rice is a variety of Oryza sativa rice produced through genetic engineering to biosynthesize beta-carotene, that is vitamin A. Main purpose is to provide provitamin A to third world developing countries where malnutrition and vitamin A deficiency predominates. The main aim of selecting rice is, as we know, rice is the staple crop of most of the world's population. The other crops like vegetables or any fruit crops which are rich source of vitamin A cannot be affordable by all the people of developing and underdeveloped world, but most of the people can afford rice. That is the reason behind why to fortify beta carotene is one of the reason to select rice. Classification of rice. The common name is Asiatic rice, belong to kingdom Plantae, phylum Anthophyta, is a monocot, class is Camelinids, order Poales, family Poaceae, genus Oryza, and species Sativa. The binomial nomenclature is Oryza sativa. The portion to enrich with beta carotene was endosperm of seed. Need for development. Milled rice is provitamin free. The symptoms of provitamin A free diet are night blindness, that is xerophthalmia. These are the photographs which are showing the effect of night blindness in children and adults. Malnutrition. Fetal susceptibility to childhood diseases like measles and general infection like diarrhea, respiratory diseases. Need for development. It was conducted with the goal of helping children who suffer with provitamin A deficiency. Children and pregnant women are at risk through this. In pregnant women, lactation deficiency, maternal mortality was seen up to 40%. In 2005, 190 million children, around 19 million pregnant women in 122 countries were estimated to be affected by vitamin A deficiency. It is commonly called as VAD. VAD is responsible for 1 to 2 million deaths, 5 lakh cases of irreversible blindness, and millions of cases of xerophthalmia annually. Why they selected rice? Here is the answer: because it's a global food, staple food, cultivated over 10,000 years, provides much as 80% or more than daily calories intake of 3 billion people, which is half of the world's population. Who began Golden Rice Project? Here is the answer. It was started during 1982 by Ingo Potraikas, a professor emeritus of the Institute of Plant Sciences. Peter Bayer was a professor of Center for Applied Biosciences, University of Freiburg, Germany. Founded by the Rockefeller Foundation, the Swiss Federal Institute of Technology, and Syngenta, a crop production company. Golden Rice Humanitarian Board, responsible for the global development, introduction, and free distribution of golden rice to target countries. Goals: more is what we aim for. Mutate rice plants to produce carotenoid or organic pigments, especially beta carotene, in the endosperm, the edible part of the grain. Make golden rice accessible locally, free of charge to farmers who are able to grow, save, consume, replant, and locally sell golden rice. The pathway of golden rice is first IPP, from there geranyl diphosphate. In that, phytoin synthase enzyme works, which produces phytoin. From there, phytoin desaturates works and keratin desaturates. These two enzymes work to produce lycopene. Then lycopene beta cyclase helps to produce beta carotene. That is a vitamin A precursor. The complete vitamin A pathway, the enzymes were taken from daffodil. Two enzymes were taken from daffodil genes and one from the Erwinia bacteria. They are given here completely. Technology that was introduced through genetic engineering by agrobacterium mediated transformation. In the first step, the plasmid is removed from the bacterium and tDNA is cut by restriction enzyme. Then the foreign DNA is cut by the same enzyme. The foreign DNA is inserted into tDNA of plasmid. Then recombinant TI plasmid was constructed. From there, the plasmid is reinserted into the bacterium in the fourth step. Then in the fifth step, bacterium is used to insert the tDNA carrying foreign gene into the chromosome of a plant cell. Then insert the tDNA carrying foreign genes, and then the plant cells are grown in cultures, and the successful plants are generated from a cell clone. All of its cells carry the foreign genes and may express it as a new trait. This is the complete pathway how beta carotene is enriched in the endosperms of rice seed. Gene transferred are three major genes: PSY, that is phytoin synthase from daffodil; CTRL, that is phytoin desaturase responsible enzyme taken from bacteria Erwinia iridovora; then ICY, that is lycopene beta cyclase taken from daffodil. That means two genes were taken from daffodil and one CTRL from bacteria Erwinia iridovora. Production of golden rice. This is the enzymatic and the operon construct where different enzymes and gene expressions were the cassette was produced. How does this golden rice works? Addition of two genes in the rice genome will complete the biosynthetic pathway, that is phytoin synthase PSY derived from daffodil and lycopene cyclase CRT1, that is from soil bacterium Erwinia iridovora, produces enzyme and catalysts for the biosynthesis of carotenoid for the endosperm. Presence of provitamin A gives rice grains yellowish orange color, thus the rice was named as golden rice. Golden rice 2. When golden rice 1 was produced, it was having only less content of beta carotene, that was 1.6 mg per gram. So that was considered as insufficient in order to meet the hunger of beta carotene. So during 2005, the team of researchers from Syngenta produced a variety golden rice produced golden rice 2. They combined phytoin synthase gene from maize with CRT1. They took phytoin synthase gene from maize instead of daffodil, and they have taken CRT1 from original golden rice instead of taking from Erwinia iridovora. That gave rise to golden rice 2, producing 23 times more carotenoid than golden rice 2. Then golden rice 1. The golden rice 2 was producing up to 37 micrograms per gram of beta carotene. Here is a clear differentiation between the normal rice, golden rice 1 and 2. This is normal rice. The light yellow colored is golden rice 1 and dark yellow or golden colored is golden rice 2. Advantages of golden rice: it provides provitamin A, easy distribution when released to needy, cheaper option to supply vitamin A requirement compared to other supplementary measures, sustainable option as once released for common cultivation can be cultivated in every growing season by farmer saved seeds, therefore no need for yearly budgetary investments for distribution. In golden rice project, what aim we have to look for: mutate rice plants to produce carotenoids or organic pigments, specifically beta carotene in the endosperm, the edible part of grain. Make golden rice accessible locally, free of charge to farmers who are able to grow, save, consume, replant, and locally sell golden rice. The research was conducted with the goal of producing a fortified food to be grown and consumed in areas with a shortage of dietary vitamin A that causes a deficiency which is estimated to kill 6,70,000 children under the age group of 5. Golden rice differs from its parental strain by the addition of 4 beta carotene biosynthetic genes. Studies have found that golden rice poses no risk to human health, and multiple field tests have taken place with no adverse side effect to participants. But disadvantages on health are may cause allergic or fail to perform desired effect. The supply of golden rice does not provide substantial quantity as the recommended daily intake. On environment, loss of biodiversity as it is genetically modified one, so uniformity may cause loss of biodiversity, may become a gregarious weed or endangers the existence of natural rice plant. Genetic contamination of natural global staple food. Then on culture, some people prefer to cultivate and eat only white rice based on traditional values and spiritual benefits. Controversies about golden rice. On August 8, 2013, experimental plot of golden rice being grown in the Philippines was uprooted by protesters. Mark Linas was a famous farmer who was anti-GMO activist. Second controversy: many parts of the world, including India, there were protests against golden rice. Cartoons against golden rice. It is all about golden rice testing. Challenges in the future about golden rice: resynthesized through latest technology, then gene transfer to tropical local varieties and targeting different varieties, level of expression of beta carotene increasing to moderate level, not too high or too less. Technological improvement, nutritional studies are essential, then make golden rice accessible locally, free of charge to farmers who are able to grow, save, consume, replant, and locally sell golden rice. The main problem with golden rice too was they were having some misconception of containing anti-sterility or causing sterility problems. So it has to be thoroughly tested, and the other challenges has to be met. Research or organizations which were involved in the research of golden rice were University of California, Rutgers University, Nebraska, and other universities. Concluding golden rice, it was said that the first recommended DNA technology crop that was unarguably beneficial. Golden rice is the brainchild of Professor Ingo Potraikas and Peter Bayer, who in the collaborative effort were able to show that production of beta carotene could be turned on in rice grains using a minimum set of transgenes. It was the first lesson to give a basic idea how the cassette of promoter or the cassette generated or the gene construct which can switch on and switch off the biosynthetic pathways which are helpful for the human or give some nutritional values. These are some of the references. Hope you liked this video. If you liked, don't forget to subscribe my channel. Thank you.